All right, guys. Hi, it's Miss Simon, and let's go ahead and get started with mutation. So go ahead and pause this video and copy down these definitions before you move on to the next thing. Okay, now that you've copied down your definitions, let's go ahead and let's see what they exactly mean. So we have something that's called a mutation. So a mutation, if you recall, is going to be something that, so a mutation, if you remember, is something that is any change in a DNA or RNA sequence. Okay, so here are all of our examples down here of our DNA sequences, changes that can happen. So let's start out with the point mutation. So a point mutation is going to be changing one base into another. What exactly does that mean? That means that could be something like changing the UUA, leucine, into UCA, which if you look at your codon chart, UCA corresponds to the amino acid serine. Okay, so I just changed one base into the other. Okay, next we have a frame shift mutation. So we know that we read our bases in groups of threes because why do we do that? Well, because that is how your codon chart works, right? You have your first base, your second base, and your third base. Every three bases corresponds to one amino acid. So that's what our reading frame is. Now, if something happens where something was inserted, like let's say instead of UUA, we now have UUGA. Obviously, if we read them in groups of three, then we know that everything behind this, right, in our DNA sequence, let's say if there's a T, C, or whatever, well, there wouldn't be a T because it's this RNA. If there was an AAA, then it's going to now suddenly shift all of those backwards, okay? If that doesn't make sense to you right now, don't worry. You're going to have some practice with it on the back page, and you'll be able to see exactly what a frame shift mutation is, okay? So again, frame shift mutation just changes the reading frame so that you are out of frame. That means it changes all amino acids after it, okay? Next, we have a missense mutation. A missense mutation. A missense mutation is something that's going to make a different amino acid. So if our original DNA sequence was UUA, which made leucine, well then our a missense mutation would be something like, let's say it's making GUA, which makes the amino acid valine. Obviously, leucine and valine are two different amino acids, which is why you call that a missense mutation. Cool. The definition for silent mutation is that a silent mutation makes the exact same amino acid. So UUA could make a mutation that turns that first U into a C, so CUA. If you look at your codon chart, CUA still makes leucine. So that's why it's called a silent mutation, because even though we had a change in the base, right, there was a mutation, a change in the base, it still ends up making the exact same product. So we call that mutation to be a silent mutation. Last but not least, we have a nonsense mutation. A nonsense mutation is something that's going to make a stop amino acid. Okay, so if you look at your codon chart, which I'll scroll down just to show you, we have a couple stop codons. See here, here, and here. So if a mutation makes it into any one of these three, that is what's called a nonsense, a stop mutation. So an example of that would be going from UUA to UAA. See that mutation, that change? That makes a stop codon, which tells us that is a nonsense mutation. Okay, so again, I'm just going to go through them. I'm going to go through and just highlight really quick the changes. We inserted a base here. For frame shift mutation, for missense mutation, we changed. And for the silent mutation, it ends up changing this first base. And then a nonsense mutation changes like this. Okay, good. So hopefully you guys have gotten all of your notes done on that part. So I have a question. Would a mutation in a sperm cell be passed on to the offspring? Why?